Imagine finally taking a pill to quiet your anxious racing mind, only to realize that exact same medication had somehow flipped a switch inside your head.
Right, a switch that just turns on a blaring, unstoppable siren in your ears.
Exactly. You try to treat the mental distress, and in return, you get this physical phantom noise that simply will not turn off.
It really is the ultimate medical paradox. I mean, we usually approach treating the brain with this comforting expectation of, isolated cause and effect.
Like pushing a specific button.
Yeah, exactly. You drop a chemical in to fix a mood and you expect it to only touch the mood. But neurochemistry doesn't respect those neat artificial boundaries at all.
It's messy in there.
Highly messy. The reality inside your skull is a massively entangled web where emotional regulation and sensory processing, they actually use the exact same hardware.
And today's deep dive is mapping that exact collision course. We are digging into some seriously groundbreaking research today, looking at a baffling biological mystery.
It's a fascinating one, too.
It really is. So we're pulling from a Science Daily article dated May 15, 2026. It covers work out of Oregon Health and Science University, or OHSU, and Anhui University in China.
Right, and this was recently published in the Proceedings of the National Academy of Sciences, right? PNAS.
Yep, that's the one.
Yeah.
And the mission of our deep dive today is to figure out how serotonin, the superstar neurotransmitter we all know for fighting depression, has basically been caught red-handed.
Caught hardwiring, a really distressing, persistent ringing into the ears of the patients who desperately need the medication.
Okay, let's unpack this, because this totally upends how we think about side effects.
Oh, completely. It completely upends our traditional understanding of how pharmacological treatments interact with sensory systems.
Right.
We aren't just looking at a story about a frustrating side effect today. The work coming from Dr. Zhenquan Tang and Dr. Lawrence Trussell is fundamentally a cartography project.
A cartography project, like map making.
Yeah, exactly like map making. They are mapping these hidden neural highways that prove just how interconnected our biological systems really are.
Well, before we get into the microscopic mapping of those highways, we have to talk about the sheer scale of the collateral damage here.
The human element.
Right, the human element, because tinnitus is not some rare anomaly. 14% of the global population experiences it.
That's a staggering number when you actually think about it.
It is. That's over 1 billion people walking around with a ringing, buzzing, or hissing in their ears that has absolutely no external source.
And, it runs a massive spectrum.
Yeah, for some, it's just a faint hum they only notice at night when the room is quiet.
But for others, it is this deafening roar that shatters their ability to sleep, to concentrate, or even just exist in peace.
And the psychological toll of that severe end of the spectrum, it cannot be overstated. A relentless phantom noise naturally triggers a profound stress response.
Oh, absolutely. It generates chronic anxiety and severe depression.
Which feeds directly into the vicious cycle at the heart of this research. Because, think about it, what is the frontline medical intervention for someone suffering from severe anxiety or depression?
You prescribe an SSRI, a selective serotonin reuptake inhibitor.
Exactly. These patients are handed a lifeline to boost their serotonin. But as soon as the medication kicks in, a terrifying number of them report that the volume of their tinnitus just dials up to 10.
And understanding the mechanics of that requires looking at how an SSRI actually alters the brain's environment.
Right, because it doesn't just magically create happiness.
No, not at all. The medication doesn't manufacture new serotonin. What it does is it blocks the reabsorption ports.
The reuptake transporters, right?
Yes, the transporters on the presynaptic neurons. So when your brain naturally releases serotonin into that microscopic gap between neurons, the synaptic cleft, the SSRI prevents it from being vacuumed back up.
So it just kind of sits there.
Yeah, the chemical pools in that gap. It continues to bind to receptors, which effectively amplifies the serotonin signal.
It feels like trying to fix a drafty house by turning up the furnace, only to realize the heat is somehow making the smoke alarms go off.
That is a perfect way to describe it.
But the problem with that pooling is the sheer lack of physical boundaries, right?
Yeah.
You take a pill orally, it metabolizes systemically, and it crosses the blood-brain barrier everywhere at once.
Everywhere. It doesn't just pool in the prefrontal cortex or the amygdala where you actually want it to lift your mood.
Right, it floods the entire system.
Yeah.
But here's the question that has always bothered me about the clinical history of this issue.
What's that?
Doctors have known for decades that SSRI patients were complaining about worsening tinnitus. Did the medical community actually believe the medication was physically causing the noise, Or did they just assume a depressed, anxious patient was, hyperfixating on a ringing that was already there?
This raises an important question, and it's one of the most frustrating parts of this history. Historically, there was a tremendous amount of clinical skepticism.
Really? They just didn't believe him.
Well, it points to a major flaw in how we validate lived experiences in medicine. When a patient says, "My antidepressant makes my ears ring," and the physician looks at the textbook,
And sees no connection.
right, they see no known anatomical wire connecting mood regulation to the auditory nerve. So the default assumption is psychosomatic.
Wow. So they just brush it off as anxiety.
Exactly. The doctor assumes the underlying anxiety is simply lowering the patient's psychological threshold for background noise. Without a physical mechanism, without the how, the biological reality of the side effect is often just dismissed.
Well, that dismissal ends today with this research.
Yeah.
Because Kang and Trussell didn't just look at clinical surveys, right? They went hunting for the actual biological wire.
And they found it. They built on earlier work from 2017, and they utilized a technique called optogenetics.
Which might be one of the most brilliant yet slightly terrifying tools in modern neuroscience.
Yeah.
I mean, we're way beyond just wiring tiny electrodes to a mouse's head here.
Oh, we are leaps and bounds beyond that. Optogenetics completely bypasses the clumsiness of traditional electrodes.
So how does it actually work?
It's wild. It's a technique where researchers use viral vectors, which are essentially deactivated viruses, as microscopic delivery trucks.
Okay, delivery trucks for what?
They program these vectors with specific genes borrowed from light-sensitive algae.
Wait, algae? Like from a pond?
Yes, literally algae. The virus is injected into the brain, where it infects only a very specific targeted population of neurons. In this study, they targeted only the neurons responsible for producing serotonin.
So you're essentially hacking the neuron's source code.
That's exactly what you're doing.
You are forcing the serotonin-producing cells to grow these microscopic light-sensitive ion channels on their surface. And once those are installed, you implant a tiny fiber optic cable into the brain.
And the precision of this is just astonishing. When you pulse blue light through that fiber optic cable, it hits those modified ion channels.
And they react to the light.
They snap open, positive ions rush into the cell, and the neuron fires. The researchers can literally command the serotonin system to fire on demand.
With millisecond precision.
Yes, simply by turning on a flashlight inside the brain.
That is mind-blowing. Okay, let's look at the actual experiment then. They take these mice, they pulse the blue light, they force the serotonin neurons to fire.
And suddenly, the auditory centers of the mouse's brain light up.
They bypass the ears entirely. But wait, here's where it gets really interesting but also a bit confusing.
Okay, what's confusing?
I have to push back on the implications of the behavioral testing here. I understand you can see the auditory cortex light up on a brain scan,
Sure, the physical scan is clear.
but the researchers claim they proved the mice were actually experiencing tinnitus. How do we know a mouse has tinnitus if it can't tap on the glass and tell us its ears are ringing?
That is a great pushback, and it's where the modified auditory startle test becomes crucial.
The startle test.
Yeah, specifically a mechanism known as gap prepulse inhibition.
Okay, break that down for me.
If you put a mouse in a perfectly quiet room and suddenly play a loud noise, it exhibits a massive startle reflex. It jumps.
Makes sense. I'd jump too.
Right. However, if you play a constant quiet background hum in the room and then briefly mute that hum just a fraction of a second before the loud noise.
Like a tiny moment of silence.
Exactly. That tiny gap of silence acts as a warning. The mouse senses the gap, it braces itself, and its startle reflex is significantly reduced.
Because it anticipated the sudden change.
Yes. But what happens if the mouse has tinnitus? If there is a constant ringing being generated inside its own head, the external gap of silence doesn't register.
Because the internal noise fills in the gap.
Exactly. Therefore, the mouse doesn't get the warning, it doesn't brace itself, and it startles just as violently as if there had been no gap at all.
That is incredibly elegant.
Yeah.
By tracking the severity of the startle reflex when there should have been a warning gap, they can definitively map whether the mouse's internal acoustic baseline has been altered.
And the data they got was undeniable. According to Dr. Trussell's findings, the moment Dr. Tang stimulated those serotonergic neurons with light, the mice lost their ability to detect the silent gaps. Their behavior perfectly mirrored an animal suffering from a continuous internal phantom noise. The brain scans match the behavior, isolating the serotonin as the sole culprit.
Which brings us to the actual anatomy of this hidden highway they discovered. When that serotonin is released, where exactly is it going to cause this chaos?
The research isolates a very specific region called the dorsal cochlear nucleus.
The DCN.
Yes, the DCN. It acts as a sort of grand central station for auditory processing in the brainstem. It is the first stop for signals coming directly from the auditory nerve in your ear.
So it's basically a filter.
Exactly. Its job is to filter, organize, and equalize sound before sending it up to the higher cognitive centers.
And what this study proves is that there is a direct hardwired circuit from the brain's serotonin factories straight into the DCN.
A direct line.
So when a patient takes an SSRI and that serotonin pools in the synaptic clefts, it isn't just lifting their mood, it is soaking the DCN.
It is. And specifically, it is binding to what the researchers identified as 5-HT receptors on the surface of these auditory neurons.
So what happens when it binds to those receptors?
Binding to those 5-HT receptors causes the auditory neurons to depolarize, meaning they become hyperexcitable. The serotonin lowers their threshold for firing.
So they get trigger happy.
Very. Suddenly, these cells in the DCN start firing off auditory signals to the brain, even when there is no actual sound coming from the ear.
It essentially creates a short circuit, generating
Yes.
a false signal that...
That the higher brain interprets as a high-pitched whine.
Okay, so if we think of the brain as a massive complex server rack, psychiatrists have basically been pouring serotonin over the whole rack to fix a software bug in the mood processor.
That's a great analogy.
And Tang and Trussell just proved that doing so physically shorts out the audio card.
Yep, it fries the audio card. But proving the cause is only half the battle. The most vital part of Dr. Tang's work in this paper is what happened when they reversed the process.
Right, because finding the circuit gives you a target.
What's fascinating here is that the researchers demonstrated that by specifically inhibiting that pathway, turning the circuit off at the DCN, they completely reversed the tinnitus in the animal models.
Even while the rest of the brain's serotonin system remained active.
Even while it remained totally active.
So they essentially found the specific fuse
Yeah.
in the breaker box that was causing the short circuit, and they proved they could just flip that one switch off.
Exactly. They turned off the noise without turning off the mood benefits.
But for the listener right now who is on an SSRI and hearing that ringing, what are they supposed to do? I mean, they can't exactly install a fiber optic switch in their own head.
No, they can't. And Dr. Trussell addresses that immediate clinical reality head-on. The application of this research today, right now, is a fundamental shift in the doctor-patient dialogue.
How so?
It is about what he terms a delicate balance. For current patients, there is no magic bullet that isolates the auditory system just yet.
So it's a compromise.
It is. The goal requires an active, nuanced partnership with the prescribing physician to heavily monitor dosage, or perhaps transition to a different class of antidepressants that interact differently with the reuptake process.
But at the very least, it validates the patient. A doctor can no longer brush this off as anxiety hyperfixation.
Exactly. The science demands that the physical interaction be treated as a primary variable in the patient's care plan.
You need the psychiatric relief, but you cannot sacrifice the patient's sensory sanity to get it.
The era of treating medication-associated tinnitus as just a secondary acceptable loss is over.
Which is huge.
It is. But the true promise of this research, and honestly, the reason the National Institutes of Health supports these kinds of investigations, even if the findings don't necessarily reflect official NIH views, lies in the future of pharmacology.
Right. So what does this all mean for the future of pharmaceuticals?
Well, Dr. Trussell envisions this as the foundation for an entirely new paradigm of drug development.
The ultimate goal here is region-specific pharmacology. Because right now, taking an SSRI is like dropping a powerful dye into a river. It colors everything downstream. You lose all control.
Right, you flood the whole valley just to water a single crop.
But in the future, they want to build a piping system that delivers the water only to the crops that need it, completely bypassing the auditory system.
If we connect this to the bigger picture, that soup model of the brain, the idea that you just pour a chemical in and let it slosh around, is a blunt instrument.
It's outdated.
Exactly. What Tang and Trussell are pointing toward is precision engineering. The future lies in designing synthetic molecules that don't just elevate serotonin globally.
They'll be targeted.
Highly targeted. They will be engineered to only unlock specific receptor subtypes located in the brain's mood centers, while actively bouncing off the 5-HT receptors in the dorsal cochlear nucleus.
And the compromise is dead. A patient will never again have to choose between managing their depression and living with a quiet mind.
That's the dream. And expanding on that thought, this research fundamentally recharacterizes how we view neurotransmitters themselves.
How do you mean?
Well, we have culturally branded serotonin as the happy chemical.
Right, you see it on t-shirts and coffee mugs.
Exactly. But serotonin doesn't know it's supposed to make you happy. It is merely a biological key.
Oh, that's interesting.
When it unlocks a door in the amygdala, you feel a sense of well-being. When it unlocks a door in the brainstem, your ears ring. It is just a utility worker doing vastly different jobs depending on the department it walks into.
We really started this deep dive with a maddening paradox that felt like a betrayal of medical science, didn't we? The idea that the pill meant to save you could simultaneously torture you.
It's a scary thought.
But by following Tang and Trussell's map, diving into those synaptic gaps, and tracing the light-activated neurons of a mouse, that paradox dissolves into clear mechanistic biology. There is no magic. There's just wiring.
Wiring that we are finally learning how to read. The discovery of this direct pathway from the serotonergic system to the auditory brainstem is a monumental leap in mapping the human connectome.
So to you listening right now, if you've ever taken an SSRI or any powerful medication and felt a strange, unexplainable shift in your body,
Like a hum in your ears.
or a change in your vision, or just some bizarre physical symptom that wasn't on the warning label, you were not imagining things.
You weren't crazy.
No. Your biological hardware is deeply structurally interwoven. A chemical cascade in one hemisphere can so easily ripple into the sensory processing of another.
Which leaves a fascinating, almost unsettling question to consider as we move through our days.
What's that?
If a molecule universally known for stabilizing our emotional state can secretly hardwire a phantom noise into our ears, what other everyday sensory experiences are quietly tied to the shifting tides of our brain chemistry?
Oh, wow.
Think about it. The way your coffee tastes sharper on a morning when you feel optimistic, the way a brightly lit room feels painfully dull when you are gripped by anxiety.
Or how we perceive physical temperature.
Exactly, or the texture of our clothes.
It forces you to wonder how much of the physical world we experience is actually objective, and how much is just our emotional chemistry silently adjusting the dials on our sensory equalizer.
It's a lot to think about.
It really is. When you pull one thread in the brain, the whole tapestry shifts. Keep questioning those connections and keep pulling the threads. We'll see you on the next deep dive.